Okay, so welcome everyone to this, I guess, the first Global Diabetes Journal Club of 2022. And it's nice to see so many people joining in. Today we have a presentation by Dr. Alisa Kjaergaard, and she is a medical doctor specialized in clinical biochemistry and a research associate from the Steno Diabetes Center Aarhus in Denmark. And today she will give a presentation about Mendelian Randomization and the nice catchy subtitle of causal inference without intervention. And so this will be sort of an introduction to this topic. So yeah, very much looking forward to it. And you go ahead, Lisa.
Thank you, and thank you very much for this opportunity to give a talk at this today's Global Diabetes Journal Club. My talk is about my favorite topic, Mendelian Randomization, or MR for short. So MR is a method that uses the observational epidemiological data and incorporates the special properties of genetic variants to strengthen causal inference without intervention. So you have probably heard or read that most published research is wrong. Numerous observational associations, although seemingly robust, have failed replication in randomized controlled trials, or RCTs. Just think of vitamin E and HDL cholesterol, both of which were suggested to be beneficial for cardiovascular health. They have been shown to be just innocent bystanders. And this is partly due to observational studies being prone to reverse causation and confounding. And true causality can still only be assessed by RCTs, as these inherently circumvent both. So luckily, MR method using observational data resembles RCTs and can be seen as nature's own way of performing randomized trials. So instead of treatments and placebo, MR approach uses genetic variants associated with lifelong exposure or interest. The Mendelian refers to Mendel's laws of heritability. So the law of segregation refers to genotype being established at conception. And since outcome cannot change the genotype, reverse causation is unlikely. Likewise, the law of independent assortment refers to different traits being inherited independently of each other. For example, genetic variants associated with high BMI are inherited independently of genetic variants associated with smoking, physical activity, etc. Therefore, the potential confounders are evenly distributed across genotype, thus mimicking the random distribution of confounders by randomization in RCTs. So because MR largely circumvents reverse causation and confounding, this allows for causal interpretations of MR study results. So the level of exposure is influenced by both genetic and environmental factors. And MR exploits the component of exposure which is influenced by the genetic variant to examine the effect of genetically predicted exposure on the outcome. MR approach relies on the notion that many environmental effects can be mimicked by genetic variants. For example, many of the LDL cholesterol lowering drugs, such as statins or PCSK9 inhibitors, which have been tested in long-term RCTs, have genetic proxies in the genes that those drugs target. For example, HMGCR gene for statins and PCSK9 gene for PCSK9 inhibitors. When estimating the causal effects of the exposure of the outcome in MR, we rely on a fundamental assumption called the gene-environment equivalence. For example, the environmental equivalent to genetically predicted BMI may be diet or exercise. However, as the genetic variants associated with BMI in the MR approach reflect the effect of lifelong BMI, whereas the weight loss in the RCTs reflect a short-term BMI intervention that happens later in life, the causal MR estimates may be larger than the RCT estimates. Indeed, for LDL cholesterol lowering drugs, the RCTs produce about 40% of the effect predicted by the MR studies. And this is not surprising as RCTs reflect a short-term intervention of 5 years or so, while the genetic variants reflect lifelong effects of the exposure. So a traditional MR analysis comprises of 4 steps. Step 1 is the observational association between the exposure and the outcome, the X and Y. The bidirectional arrow illustrates the uncertainty about the causality. Step 2 is the instrument exposure association, and step 3, the instrument outcome association. Thus, the step 3 is an indicator of potential causality. If genetic variants associated with the exposure also are associated with the outcome, then this suggests that exposure is a causal factor for the outcome. In a full MR analysis, step 4 calculates the magnitude of the causal effect of the exposure on the outcome. So these genetic variants or instruments, what are they? Genetic variants mostly used in MR studies are single nucleotide polymorphisms, or SNPs. SNPs are common DNA sequences, sequence variations that occur when a single nucleotide, A, C, T, or G, in the genome sequence is altered. In this example, we have a C to a T SNP, where the C allele has a frequency of 79% and the T allele a frequency of 21% in population. Since we inherit a set of genes from each parent, possible combinations are 2 Cs, 2 Ts, or 1 of each. So if the T allele is associated with a higher BMI than the C allele, then the T allele is the effect allele with regards to BMI. This slide shows allele frequencies on the x-axis and the effect sizes on common diseases on the y-axis. And most genetic variants fall into the middle diagonal part of the graph. Common variants usually have weak effects on common diseases, while more rare variants may have stronger effects. Most common variants implicated in common diseases are therefore identified by the so-called genome-wide association studies. And what are these? Well, exactly what the word states. Association studies of the trait examined across the entire genome. Let's say we have a group of individuals with type 2 diabetes. These are cases, and a group of individuals without type 2 diabetes. These are controls. After performing a massive genome-wide genotyping, a regression analysis of each SNP on type 2 diabetes is performed to examine the SNP type 2 diabetes association. And the result is shown as a Manhattan plot at the bottom, where association with the type 2 diabetes is shown for each SNP according to the chromosomal position on the x-axis, and the y-axis shows the strength of the association, the minus log p. So the p-value here of 5 times 10 to the minus 8, which is the top dashed line, is considered to be GWAS significant. And if you can think of that as a p-value of 0.05 divided by 1 million. And here on the Manhattan plot, spikes represent genetic loci potentially associated with the trait examined, such as type 2 diabetes. And of course, in these analyses, continuous traits are examined by linear and binary traits by logistic regression. This slide shows the conceptual illustration and the 3 core, that's gone. So the 3 core assumptions of the MR method. So the first assumption is the relevance assumption. This assumes that genetic variants are strongly associated with the exposure. But how are these genetic variants identified? How can this be done? So in a targeted approach, we can use our biological knowledge and function. I have done this previously with the plasma biomarker called YKL40. This YKL40 biomarker has a promoter SNP, which is situated close to the gene for YKL40, and it has a known functional significance. However, for many other complex diseases, such as smoking initiation, there are no obvious single genes to use. So here we would use a shotgun approach, which translates into using GWAS. And when we use GWAS, then most of the hits are actually non-coding regions with unknown function, as illustrated below. And this can give us a bit of headache when interpreting the results. The second assumption is that the genetic instruments are not associated with confounders. This is usually not an issue. The only real confounders here can be population stratification. So that means population differences that relate to different distribution of genetic variants and different distributions of the outcomes in different populations. And this is why most MR studies to date are performed on European populations only. The third assumption is that genetic instruments are associated with outcome through exposure only, meaning that the entire effect on the outcome is mediated through the exposure. The violation of this assumption is known as horizontal pleiotropy. That is, the genetic variant has an effect on the outcome through a completely independent pathway than the exposure. And this is probably the biggest challenge in the MR approach. So, in repetition, the magnitude of the causal effect of X on Y is estimated in step 4 of the MR analysis. Step 4 estimates the effect of genetically predicted X on Y, that is, the causal effect of X on Y. In practice, this translates to a two-stage least squares. So first stage is the regression of instrument on the exposure, and the second stage is the regression of outcome on genetically predicted exposure from the first stage. So this is why we get the effects of genetically predicted exposure on the outcome. And this, of course, applies to continuous exposures and outcomes because linear regressions are applied. In case of binary outcomes, such as type 2 diabetes, the second stage is replaced by the logistic regression of the outcome on genetically predicted exposure, which also includes the residuals from the first stage. And this approach is called control function estimator. Now, these approaches were used in this study, examining, among other things, the effects of BMI on the risk of type 2 diabetes. So table 2 shows the observational estimates, and table 3, the MR estimates. MR estimates were higher than observational estimate, which is not that surprising, as I mentioned earlier. And as you see here, the authors used the two-stage least squares analysis for continuous outcomes, in this case, probably blood pressure, and control function estimator for binary traits, such as type 2 diabetes. But they also used the polygenic risk score method, which employs the WALD ratio estimator. And what is this? So the WALD ratio estimator is the ratio of the instrument outcome and the instrument exposure association. So the causal estimate, shown with the black arrow, is simply calculated as the instrument outcome association, shown with the blue arrow, step 3 in the full MR analysis, divided by the instrument exposure association, shown in the red arrow, which is step 2 of the MR analysis. So again, in repetition, a traditional MR comprises 4 steps and is performed in a single population sample with individual level data on all participants. In contrast, a 2-sample MR uses 2 population samples, one for the instrument exposure association and another for the instrument outcome association. And here only summary level data, such as those publicly available from published GWAS, are needed. For example, if we wanted to examine the causal effect of BMI on type 2 diabetes, we would use the summary statistics from a GWAS on BMI in one sample and from a GWAS on type 2 diabetes in another sample. This study investigated the causal effects of smoking initiation on the risk of type 2 diabetes using a 2-sample MR approach. The instrument exposure data were from a GWAS on smoking initiation in 1.2 million individuals, and this GWAS identified 377 SNPs associated with smoking initiation. And the instrument outcome data were from GWAS on type 2 diabetes in 900,000 individuals. So from smoking GWAS, the summary stats for the 377 smoking SNPs were extracted, shown on the left, and aligned to the summary statistics of the same 377 smoking SNPs in the type 2 diabetes GWAS. And note here that the effect allele is aligned. So for the first SNP, we had the effect allele is the T allele. And the summary statistics, the beta coefficient has to have the same direction here. Otherwise, we're messing everything up. So for each SNP, then, meaning for each row here, a WALD ratio is calculated. And this WALD ratio is the beta coefficient for the type 2 diabetes divided by the beta coefficient for the smoking initiation, meaning the instrument outcome association divided by the instrument exposure association. Once we have a WALD ratio for each of these SNPs, then a meta-analysis can be performed to calculate the combined causal estimates of smoking initiation on risk of type 2 diabetes. And here are the results. As you can see, the top row shows the IBW random effects, and this stands for inverse variance weighted random effects meta-analysis. And this is because the meta-analysis this is the standard approach for this. Other weighing methods can also be used in other sensitivity analysis. But regardless, the results here showed that genetic liability to smoking initiation was associated with increased risk of type 2 diabetes. So here at the end, I would like to show you results of a 2-sample MR we recently published on obesity and kidney function. So as exposures, we considered all SNPs associated with BMI, but we also examined in detail BMI SNPs nominally associated with type 2 diabetes, BMI, type 2 diabetes, and also SNPs nominally associated with high waist-hip ratio, which we call adverse BMI SNPs. So these adverse BMI SNPs are SNPs associated with both high BMI and high waist-to-hip ratio. We also looked at waist-hip ratio alone and waist-hip ratio adjusted for BMI. Overall, the results showed that genetically high BMI was associated with low eGFR, with high blood BUN or blood urea nitrogen concentrations, and high risk of microalbuminuria and CKD or chronic kidney disease. And if you notice, the association was particularly pronounced in adverse obesity, but less pronounced for waist-to-hip ratio. We also performed a bidirectional MR and looked into whether genetically impaired kidney function is associated with obesity. And here we found no robust association in the opposite direction. So the conclusions of this study were that obesity was causally associated with impaired kidney function and not vice versa. And the effect was particularly pronounced in adverse obesity, meaning both high BMI and high waist-to-hip ratio. So to examine direct and not only total effects of obesity and kidney function, we also performed multivariable MR. And this can be thought of as adjusting BMI and waist-to-hip ratio for each other as well as for type 2 diabetes. And these results show that genetically high BMI adjusted for waist to hip ratio and type 2 diabetes were causally associated with impaired kidney function, that is decreased eGFR, increased BUN, and increased risk of chronic kidney disease. However, adjusting for diabetes here abrogated the association with microalbuminuria. And this is interesting because in our MR analysis, genetically high BMI was associated with increased urinary albumin to creatinine ratio in populations with diabetes, but not in the general population. Now, this suggests that BMI, unlike waist to hip ratio, was directly causally associated with impaired kidney function. But the causal pathway to albuminuria was likely through type 2 diabetes. And I guess I'm done with my presentation. I'm not sure if I actually talked too fast or didn't have enough slides, but having 35 slides, I thought I should stop and was expecting this would take too long, but apparently not. So
no, this is I think this has been great and a good pace and a good explanation. So no, thank you very much. I think this was really great, and I think you did a very nice job at explaining some things, and I actually got some new terms with me also, sort of to understand sort of what or put some more words to what is actually some of the concerns that one could have.